Okay, so today's uh, lab is going to be the respiratory lab. So let me get into my share. Okay, so this has a lot of structure functions, just like the heart did. And same with identifying different layers of tissue and what tissue would be found in those layers and their functions, and then some physiology toward the end. So part one here, the respiratory anatomy, using available models as your guide, identify and describe the functions of the following respiratory organs, which are mostly captured in the matching down below. All right. Okay, so when it comes to the images, and we will first look at the images contained here in the web page, but also use the PowerPoint that I put up there, because that has a lot of pictures that I think are even sometimes better than what is on the course page. And for that particular lab this week, and it will identify more structures. So those are very good pictures to use to study from as well. So we're going to look at the nares, the nasal cavity, paranasal sinuses, the pharynx, which is the throat, the nasopharynx, oropharynx, and laryngopharynx. The larynx, the parts of the larynx are actually the thyroid, cricoid, arytenoid cartilages, and the vocal folds. And the hyoid bone and the epiglottis are on that same models, but they're not exactly part of the larynx. The trachea, with their cartilaginous rings of hyaline cartilage, and the membranous portion. We have then bronchi, bronchioles, alveoli, lungs, and diaphragm. All right, so let's look at some of these functions. And as usual, it's generally easier to start with the options and match them to the structures. And I will tell you in advance that some of these functions can be used for more than one structure, because the respiratory system is, with the exception of the pharynx, is that it has a lot of mucus secretions there. And therefore, if you have mucus, then you're trying to warm and moisten air, trap pathogens, and be a conducting pathway for air. But sometimes you just want to be specific, as to, let's say, conducting air from maybe here to here and then here to here, because that structure is found in certain particular regions. So when it's applicable like that to use it for more than one structure, I will, give you an indication of that. All right, so we have the common pathway for food and air. That would be the pharynx. All right, so the pharynx is going to be broken down into more than one region, specifically nasal, oral, laryngeal pharynxes, but A goes with pharynx. B, site of air entry to the system would be your nares or your nostrils. C, the organ that contains both conduction and respiratory structures. That would be the lungs. D, the conducting tube anterior to the esophagus that contains hyaline cartilage. That is the trachea. It transports air from the larynx down into the thoracic cavity, but not into the lungs. E, elastic cartilage flap that covers airway opening during swallowing. That is your epiglottis. F, warms and moistens air. That's primarily your sinus cavities. Remember in A&P1, we looked at the sinus cavities or paranasal sinuses, the frontal, maxillary, sphenoid, and ethmoid sinuses. And we said that they warm and moisten air, helps trap pathogens, adds resonance to your voice, lightens the skull. So there are some other functions as well. G contains the vocal folds. That would be the larynx. H traps debris and mucus secretions. Well, honestly, again, wherever you have mucus, which is in the nasal cavity, in the sinuses, in the trachea, the bronchi, really, wherever you have mucus being released, you're going to have, um, that same function. So it's not unique to any one area. But I will just say generally, as long as you give one major function for those um, structures, you'll be fine. I, conducting tube in the lungs, able to regulate airflow by constricting and dilating because they contain smooth muscle. Those are going to be the bronchioles. Just like an arteriole, you have the smooth muscle that is capable of vasoconstriction dilation. The bronchiole will do the same thing. Therefore, it's under the control of the autonomic nervous system, the sympathetic nervous system versus the parasympathetic division. And so I would review what are my my sympathetic and parasympathetic airway responses. In sympathetic mode, you want to bronchodilate, and you want to let more air into the system, reducing the flow of air. And in parasympathetic stimulation, you want the bronchos constrict. Right, J, add resonance to voice. That would be the sinuses as well. K, conducting tube that transports air from the carina to the lungs. The carina is where it is found at the inferior aspect of the trachea, where it branches off and bifurcates into a right and left main bronchus. And so that's what we're referring to is a bronchi. Bronchi transport the air actually into the lungs. L, alters sound pitch when tension or length is changed. Those would be the vocal folds or vocal cords. Site of gas exchange with capillaries, that is the alveoli. And N, anchors the vocal folds, those would be the arytenoid cartilage, because they're by themselves, they're the arytenoid cartilage. Okay, so moving on to the histology. Identify layers and tissues and describe functions of the layers for each respiratory organ. So, we did similarly to the layers of the heart and the layers of the blood vessels. We have an epithelial layer lining the lumen of the tube. Then we generally have a subsequent layer. In this case, in the trachea, we'll have a submucosa. And then we have a hyaline cartilage, and the adventitia is the outer connective tissue layer. Right? So, and the trachea, or let's say the bronchi, is virtually identical histologically to a trachea. It just doesn't have much of a submucosa, and it doesn't really have an outer connective tissue layer per se, because it's actually within the lung tissue itself. Okay, so when we're looking at the slides, we're looking at the trachea, we have these four layers: the mucosa, submucosa, hyaline cartilage, and adventitia. The mucosa is composed of what we call PSCCET, pseudostratified, ciliated columnar epithelial tissue. So it's a row of single columnar cells that has cilia on the surface. Said pseudostratified because it appears to be multiple layers when it's just one. And it's epithelial tissue, or epithelium, because it lines the lumen. Then deep to that, or I should say really superficial, because we're radiating outward, we have the submucosa that contains areola connective tissue and supracuboidal epithelial tissue. Similar to the dermis, where you had that dense irregular connective tissue and you had a lot of supracuboidal epithelial tissue because sweat glands were found there. Well, in the submucosa of the trachea, we have a lot of mucus glands there. So that's why, we also find supracuboidal epithelial tissue. But the majority of the other connective tissue is going to be areola connective tissue. And then as we radiate outward, we have a layer of hyaline cartilage, which is hyaline cartilage. It functions to maintain a patent airway. The hyaline cartilage doesn't completely circle the whole circumference of the trachea. On the posterior aspect, we're going to have kind of this connective tissue, muscle tissue that's called the membranous portion, so that the cartilage portions are what we call C rings. They don't go all the way around like an O, or a zero. Yeah. It stops on the posterior aspect, so it forms kind of a C shape. And that way, as food passes through the esophagus, you've got a membranous portion back there that'll allow the esophagus to dilate and food not get caught on this cartilaginous rings of the trachea, so that it can be moved down to the stomach. The adventitia is a connective tissue layer. And like any outer connective tissue layer of an organ, it helps to secure it in its location. It helps to protect it from other organs adjacent to it that are moving and stretching so it doesn't become impaired by friction. It also helps secure blood vessels and nerves to that structure. Right, then we go into the lung itself. Now, we have bronchi, bronchioles, and alveoli. Just like arteries to arterioles to a capillary. We go from a large tube, like a bronchi, that is very, very, very similar to a trachea. It has a mucosa of pseudostratified ciliate columnar epithelial tissue. And then we have a very slim little region of submucosa to a cartilage ring. And then you have a little bit of connective tissue to the exterior surface of the leaf, especially at the proximal end. So the bronchi transports air from the carina out into the lungs. And again, the piscate, the pseudostratified layer, has goblet cells there, that are going to release and be all mucus lined, and they capture the material that you're trying to filter out of the air, so it's clean air by the time it gets down to the alveoli. And those cilia contract and move upward. The mucus that's on the surface, so that it moves upward, so it doesn't settle into the lungs, and that you keep cleansing the air pathway. The bronchioles are like an arteriole. They're capable of bronchoconstriction dilation. So histologically, there are some changes here. The mucosa is composed of simple cuboidal epithelial tissue. And then instead of having hyaline cartilage, it has smooth muscle. And that's the muscle that can contract and bronchoconstrict and dilate. So under the control of the autonomic nervous system. And it transports air from a bronchi to the alveoli. So now we end in the alveoli. And just like a capillary, it is a layer of simple squamous epithelium. So you'll have the wall of the alveoli, simple squamous epithelium, adjacent to a capillary wall, which is also composed of simple squamous epithelium. So as blood passes through the capillary, and those two squamous cell layers are, nice and close to one another, gases can diffuse from carbon dioxide into the blood, into the alveoli, and from in the alveoli, oxygen into the blood. And that's where we gas exchange at what's called our external respiration site to swap gases, as the blood passes through the lungs. And remember that the arteries in the lungs are blue and the veins are red. They're one of those locations of two that are the exception, that all arteries are red and all veins are blue. Remember, because we're still pumping deoxygenated blood in arteries away from the heart, but right from the right ventricle. So we are still blue vessels because we are still rich in carbon dioxide and need to gas exchange. Okay, so we'll apply those to the images in just a few minutes. We have pressure volume relationships in the lungs, indicate what each of the structures does during ventilation. So you should be able to describe what each of these contributes to inhalation and exhalation, or inspiration, expiration, and be able to identify them as many as possible on models. And that's why the PowerPoint notes actually point to more things. These are some images from a former image database that we had that lets you see more things. And then I can also point out a few of them to you. Okay, so intercostal muscles in the ribs. Intercostal muscles contract, and the rib cage move up and outward, like up and laterally, in what's called a bucket handle fashion. So they move up and out. The diaphragm contracts and moves inferiorly toward your stomach. That's why your stomach rises when you inhale. The diaphragm puts pressure there and forces some of the organs of the abdominal pelvic cavity to kind of distend. Elastic connective tissue in the lungs. Well, between the alveoli throughout much of the lungs are elastic fibers. So what happens to the elastic tissue? It becomes stretched like a rubber band. And the pleural membranes follow the diaphragm and the rib cage because they're attached to the interior surface of them. And so the pleural membranes get pulled with the rib cage and the diaphragm and pull on the lungs and try to expand the lungs, creating a negative pressure inside so that you suck in air. Exhalation is the opposite, right? Intercostal muscles relax and the rib cage moves downward and medially. So now you're compressing the lungs and the diaphragm relaxes and moves upward. So from both directions, now you're kind of squeezing down on the lungs to exhale air. Elastic tissue in the lungs recoil, so it helps collapse and compress areas in the lungs that are not closest to the ribs or to the diaphragm. And the pleural membranes, again, just follow the diaphragm and the rib cage. Okay, pH and ventilation. This is a little wet lab that we do in the lab. And we're going to talk about the results in here. There are a couple pictures in the images that are on the course page, not necessarily in the PowerPoint notes, because this has been added since then. All right, so we're going to have two test tubes of deionized water, just plain water. And we are going to measure the beginning pH by using a little piece of litmus paper and matching it up to the pH scale. And both of them basically begin at like 7.0 at neutral. Now in tube A, we have a straw that we're going to have one student take a breath in, hold for a moment, and then slowly exhale through the straw and blow bubbles in the water. In tube B, we're going to have a little straw with a bubble at the end that you're going to let the bubble fill with air. You're going to depress the straw into the water and squeeze the bubble so that you are forming air bubbles in the water. So this little device, allows you to let the bubble fill up, the bulb fill up with air, and then you compress it and you squirt air into the water. So it's similar to blowing, air through a straw, into the water, but we're not going to use air that we're breathing out because that's going to have carbon dioxide in it. So we need ambient air. And so this little bulb will do that. You place it down to the water, you squeeze it, you lift it out of the water, let go of the bulb, let the air refill with water, with air, my bad. And you just keep doing that over and over for about 30 seconds to a minute. So then you remeasure the pH. In tube A, I would expect to be about 6.2, maybe 5.8. In tube B, it would be really what you started with, about 7.0. So the gas added to tube A would be what? If we're exhaling through that straw into the water, we're delivering carbon dioxide. So in tube A, gas added is carbon dioxide. In tube E, it's just air. Because all we did was we used that little bulb to deliver and blow, create air bubbles in the water, which does not affect pH. So which tube had a decrease in pH and why? Well, that would be tube A. Why? Because we added carbon dioxide to water. And that's then part of the carbonic acid cycle that increases the hydrogen ion concentration and decreases the pH. And thus, that's why the pH was less than what we started with. Which tube did not change and why? That would be tube B, because oxygen does not affect pH. Okay, then we're going to describe how the following conditions affect blood levels of oxygen, carbon dioxide, and pH levels. So this is a good time really to look at the items in red down here. First, we have the carbonic acid cycle, which you should have tattooed on your ankle because you're going to use this for the rest of your life. Water, H2O plus CO2, yields carbonic acid, which is H2CO3, which then yields hydrogen ions, and bicarbonate ions. All right, so remember, what determines the pH of a solution? The ratio of hydrogen ions to hydroxide ions. So, when we put more carbon dioxide or we have less carbon dioxide in a solution, then we can alter the amount of hydrogen ions being produced and therefore change the pH level. As hydrogen ions increase, pH value decreases. As hydrogen ions decrease, the pH value of the solution increases. All right, so here, and we're going to come back to this and then the urinary system, because water is abundant in the body. So that's not a limiting factor. But the amount of carbon dioxide in plasma is variable. Right? So down below, we have normal carbon dioxide of arterial blood. The normal range is 35 to 45 millimeters of mercury. So we should have a certain amount of carbon dioxide normally at all times, right, in arterial blood. If that goes higher than normal, so let's say we have a carbon dioxide level of 50, that means, looking at the carbonic acid cycle, is that when we have more carbon dioxide in this system, we produce more of the acid and ultimately more of the hydrogen ions. And therefore the pH value goes down and we become, it's called acidotic or respiratory acidosis. If the carbon dioxide levels are lower than 35, then the carbon dioxide level decreases, and thus so does the hydrogen ion concentration decreases, and the pH value goes up and increases. That is what we call alkalosis or respiratory alkalosis. So the normal range of 12 to 20 should about maintain that 35 to 45 millimeters of mercury. But then we look at now part B, going back to part B. Well, what effect does hypoventilation have on these levels? And what effect does hyperventilation have on the level? So hypoventilation, think of holding your breath or anything that would cause carbon dioxide to build up in the blood. All right, so what happens to the oxygen levels? Well, the oxygen levels start to decrease because you're not taking in more oxygen, and your cells still need to use oxygen for cellular respiration. So the oxygen levels are going to start to diminish. CO2 levels increase. And if CO2 goes up, what happens to the pH level? It goes down. Right? CO2 increases, hydrogen ion concentration increases, pH value decreases. And then the opposite is for hyperventilation. Now we're blowing off too much carbon dioxide. Now it says here, hyperventilation, during exercise, but you most readily see this when you have hyperventilation of someone who's panicky, or sometimes children, and will start to hyperventilate when they're getting upset about something. Maybe you're panicking, you're afraid, and you start to hyperventilate. So when you hyperventilate, oxygen levels will either be normal or increase a little bit. Carbon dioxide levels will decrease, and the pH value will increase. Okay, and that is the last of those lessons in the respiratory system lab. I'm going to change my share and get into the images because that seems to help us coordinate these things. All right, so let's go here, and let me go to my course content, and we'll get into the lab. There is some additional information in the notes that I provided you, but let's get through the images first, and then I'll go back and hit some of those highlights. All right, so we're going to go to lab 5-1 image, and let me change my share. Okay, here it is. All right, so this is one of those cookie sheet models, I call them. This is actually a very large model. This is one like the upper left corner of this model, but it is indicating, what's that arrow 1, 2, 3, and 4. It's not pointing to the nares, which are to the way anterior aspect, but it's pointing to that region of, on the model, it's 3, 4, 5, and 6 arrows, 1, 2, 3, and 4. So we have the nasal cavity, nasopharynx, oropharynx, laryngopharynx. Image here. Nasal cavity, nasopharynx, oropharynx, and laryngopharynx. And you should know, what they're, where they're located and what kind of tissue is found there. Wherever food and air travel in the oropharynx and the laryngopharynx, and of course the oral cavity, it will be lined by stratified squamous epithelial tissue. This food is very coarse. Okay, then we're looking to kind of like the upper left-hand side of that same model. And I know these are kind of sideways, but that represents one, the trachea and the bronchi. So all those little white branching regions here at 21 and 22 are all part of bronchi. One is the trachea. And above that in blue in number 16, that represents the larynx. The larynx. So we have the trachea. I would know the layers just like you would for the slide. What is in the mucosa, the submucosa, the hyaline cartilage, the adventitia, what are their functions? Apply it to the same, to these models over and over and over again. So the trachea conduct air, from the larynx down into the chest, then into the thoracic cavity, not the lungs, but into the thoracic cavity. It's that tube that's anterior to the esophagus. It's lined with pseudostratified ciliated columnar epithelial tissue in the mucosa. And again, I would just keep repeating to yourself as if you were looking at a histological side of the trachea is, what would I find in the mucosa and the function, the submucosa and the functions, the hyaline cartilage and its function. If there's the adventitia, which is your connective tissue there and its function. And then we have the bronchi. So the bronchi are now transporting the air from where the trachea bifurcates at the corona. Now it goes out into the lungs. And it has very similar histology as the trachea. And that's one way you're going to recognize it on the slide is you see some hyaline cartilage kind of peeking out around the circumference of this tube you're going to be looking at on end. Because once you're in the lungs, you can see generally all kinds of things. You're able to see bronchi and bronchioles at the same time and the alveoli. So you need to be able to distinguish the difference between them. All right. It too is lined with pseudostratified ciliated columnar epithelial tissue. It has the cartilage there, and so that's how you in this layer, in this second layer. So that's how you're going to recognize it. All right, then we have to the lower right-hand corner on that same model. This is where what we use to identify the bronchioles and the alveoli. So 1 is the bronchiole and 2 is the alveoli. 3 is pointing to a diaphragm. So we look at the numbers on the models, 34 and 34 in black here, those are the bronchioles. 38 and 37 are just identifying the different blood vessels. The blue one would be a pulmonary artery. The red would be a pulmonary vein. Remember, because, in the lungs, the arteries are blue and the veins are red. Okay, then we're looking at the alveoli, which there are multiples. They kind of look like, grapes on a stalk. You have one stalk with multiple grapes branching off from there. And here at 36, it cuts into them. So you can see these walls of the alveoli and air in the interior. The arteries and the veins, they form a capillary network that is, adjacent and approximated to the alveoli. of the um, or the simple squamous of the alveoli. Sorry about that. Had to kill a bug. My bad. Sorry, I just had this palmetto bug walking along the wall, and I was like, "Oh my god, it has to die." My bad. I'm so sorry. Okay, so that capillary bed, the simple squamous of the capillaries and the simple squamous of the alveoli together form what's called the respiratory membrane. And those must be very, very close to one another, to allow gases to, most efficiently diffuse across that membrane, at the area of external respiration, we call it. All right, so we have a bronchiole lined with simple cuboidal epithelium. Remember, the bronchiole is going to have the smooth muscle capable of bronchoconstriction and dilation under the control of the autonomic nervous system. Alveoli are composed of simple squamous epithelium. This is our site of gas exchange. And that the diaphragm is that muscular wall is active in inspiration and expiration, or inhalation and exhalation, and know the directions it must move. All right. It goes upward during exhalation and downward in inhalation. Remember that, because I know it's kind of contrary to what you might think, but the diaphragm has to contract and drop downward. I'm going to go back to this image just for a moment, because you can also see at like 26, I believe, they're indicating ribs that are kind of cut and looking at you on end. And then you have intercostal muscles in red in between them. And then kind of traveling left from that point toward the heart, you can see these, connective tissue layers next to it. And those would be the pleura. You don't have to distinguish really between the visceral and parietal pleura, but that's where the pleura would be found. And so you can see how if the ribs move, it pulls the pleural membranes. And the same, you can see it wrap around the diaphragm. So as the diaphragm moves, it pulls on those membranes as well. That just is a good opportunity to see those on those models. Okay, then we're looking at larynx models. We have both of them here, which is nice. The gray one on the left and the one to the right is kind of greenish, like light bluish. Generally, where you see light blue on cartilage, that means it's going to be higher than cartilage. So to both of them up at the top, they're not necessarily, don't have an arrow on them right now, but those are the hyoid bones, right? The hyoid bone on both sides. On the one on the right, I think it has a number 9 on it. Right, then below that, right, we have what's called the thyroid cartilages, which is the broadest when we're looking, from anterior to posterior. We're looking dead on to your hyoid bone and like this to your larynx. That is the thyroid cartilage. And then below that would be the cricoid cartilage. Okay. This is looking at the posterior aspect. All right, so you kind of have to flip the images around, but one is pointing to the epiglottis, elastic cartilage. Right? It moves to block the entrance into the larynx when you're swallowing food, so you don't aspirate, and the food goes into your esophagus. 2 points to the only paired cartilages called the arytenoid cartilage, and they pull tension on the vocal cords or the vocal folds that are in the interior of this tube. This doesn't show you very well those, but I will say that in the gray model, if someone uses a gray model, right, or the gray model shows up, it looks like pieces of white cotton in the center, that are, like fabric. In the one to the bottom here or to the right, if you've inverted this, it's like a flesh-colored rubbery tissue on this particular model, kind of like the esophagus, but without any bumps on it. Okay. Now we have slides, and this is a slide from the trachea. We actually looked at this in AMP1 also, because this is a really good um, organ to use to find abundant hyaline cartilage. So the surface layer there, that's your mucosa. Right? And you can see in another slide in the notes, I think you can see it a little bit better, but it's a single layer, but it looks like multiple layers of columnar cells, pseudostratified ciliated columnar epithelial tissue. The layer is the mucosa. All right. Between that and the hyaline cartilage is your submucosa, and that contains areolar connective tissue and simple cuboidal epithelial tissue, because we have mucus glands there, and you can actually see some of them. Even though it's not really clear, because this is hard to take. I find it hard to take pictures through the microscope. Now what I do is I put it on the overhead, the front projector, to the in front of the classroom, and I just take a picture from my phone that way. But I do have some better ones in the notes. So we have the mucosa, submucosa, hyaline cartilage layer, which is hyaline cartilage, and to the inferior aspect where you can see the adipose connective tissue there, the soap bubbly tissue, that is where we find the adventitia, the connective tissue layer. So just because we what we cover in the lab exercises, is what you're responsible for in all different images that can be used that demonstrate that anatomy. So, we need to, use both sources, because those, will be used for quiz and exam purposes. So it says identify the layer, the organ, the tissue, what's that clear cell called? That's a goblet cell contained within it. And then the third layer is the hyaline cartilage here. And for some reason, I just don't know why he didn't indicate, the submucosa as well, but and the adventitia, but it's easy to identify those, at least those two on this slide. And then when we look at this other picture, I will point it out to you. Right? So it's the trachea, the mucosal layer, simple, pseudostratified ciliate columnar epithelial tissue. Traps pathogens, warms and moistens air. The goblet cell produces mucus, and the hyaline cartilage helps maintain a patent airway or prevents collapse. So wherever you have hyaline cartilage along this pathway, it just keeps the tube from being crushed. All right, here is a slide of a bronchi. So we know we're in the lungs. So, one of the reasons why it's a bronchi, or there's two reasons. And one is we're in the lung, because we can see, especially to the bottom left-hand corner, we can see this soap bubbly kind of chicken wiry honeycomb kind of tissue. That's the simple squamous epithelium. So if you see simple squamous epithelium alveoli, you're in the lungs. So the things that are in the lungs are bronchi, bronchioles, and the alveoli. The trachea does not enter into the lungs. So over to this, if you look at the tip of the pointer, and you go to about 5:30 from there, about 5 o'clock, 6 o'clock, you see that light staining, like light purplyish, soap bubbly material? That's hyaline cartilage. If you did not see hyaline cartilage somewhere around here, then you would call it a bronchiole. Over about 9 o'clock, there's another small pale staining lavender region as well. So this is a bronchi. You must see somewhere the hyaline cartilage there to call it a bronchi. All right, so it's a bronchi. Notice the hyaline cartilage, even if there's small areas, and it's lined with pseudostratified ciliated columnar epithelial tissue. Okay, now we get to, we're in the lungs, because I can see all these alveoli, and we have a tube, but we're not sure what it is. Is it bronchi or bronchiole? I don't see any hyaline cartilage around the periphery. And if we magnified it a little bit more, if we had a slightly better picture, which I believe I do have one or two maybe in the notes, in the PowerPoint notes, you'll see a little bit of smooth muscle in there. Then it confirms you are looking at a bronchiole. So what's different about a bronchiole to a bronchi? The mucosa is simple cuboidal epithelial tissue, and we have smooth muscle instead of hyaline cartilage that can bronchoconstrict and dilate, and thus be under the control of the autonomic nervous system. So I was asking, maybe know the action of epinephrine and norepinephrine. Well, remember, epinephrine and norepinephrine is used in the sympathetic nervous system. So the question is ultimately, what will be the response of that bronchiole if we are in sympathetic stimulation releasing epinephrine and norepinephrine? And that would be bronchodilate. So it's actually an inhibition to the impulses traveling to the smooth muscle, right, to let it relax a little bit and dilate and be widened, just like we do sort of with the arterioles that are only controlled by the sympathetic nervous system. Okay, identify the structure, tissue, function. We just did. Yeah, so alveoli, simple squamous epithelium, and gas exchange. So the alveoli is composed of simple squamous epithelium and gas exchange. Let me just go back real quick. Okay, so I just want I neglected to just strictly identify what was at the tip of the pointer. I mean, that is your alveoli. The bronchiole is the large tube passageway in the center, but those smaller ones to the side kind of looks like Swiss cheese or honeycomb appearance. Those are the alveoli, gas exchange. In lecture, you probably have to know, the type 1 and type 2 cells, and you have some macrophages there, but we don't generally ask you that here. Although it would be good to know that surfactant is released at that region to help prevent collapse of those alveoli. That would be helpful. Okay, this is the image of that wet lab portion that I was describing. So we had two test tubes, or my test tubes that contained water, and we put in pH indicator in there. And so you shake it and swirl it, and you match it up to the scale, here in the center. If anybody has a fish tank at home, you probably do this all the time. And so it's about neutral. It's a little higher than that, but let's say it's starting at about neutral. And so starting at the blue range. Tube A has the straw that you're going to blow into, and B has that little dropper. You can't see the top of it, but it's a little straw with a little bulb at the end that can air fill. And then you let you put the end of the dropper or the straw into the water, and you squeeze the tube, and it forces air out. So you produce little air bubbles in the water. So that's before we add anything to the system. And then after, all right, we blow into the straw, we just squeeze air in. You almost see the little bulb there at the top of that dropper. A changes to a green color. So that's because you added the carbon dioxide there and it became acidotic. The B, you just added air to, so the pH never changed. And that comes down to the carbonic acid cycle. You can skip over these. I cover these in lecture, but I don't cover them in lab. So for all intents and purposes for us, at least right now, we're not covering a lot of the pulmonary volumes, but I do cover them in lecture. When appropriate, that actually is, there's a kind of a fun way to measure those. You can do water displacement exercises that let you measure, as you exhale through, not a straw, but it would be a larger tube, and you have a system of water, in a tall flask that, as you blow in the water, it displaces. As you blow in air, it displaces the water upward, and you have like a little ping pong ball or something on top, so you can see the level changing, and so it measures how much you're moving from your lungs into the system, and then you measure them. You measure your your tidal volume, which is the normal air, the quantity of air that you're breathing in and out, turning over in your lungs, just normal breathing at rest, and then you have your expiratory reserve and your inspiratory reserves. So, but just looking at the pictures, I do that in lecture. Okay, so let's quickly just look at the notes because I don't want to keep you too long because you start generally to kind of get lost in all this. But I have the PowerPoint notes here, and we're going to just kind of zip you through to some images, by especially. You can read through here because these sometimes is a little more than what you need, but it talks about structure, function, of virtually everything you have to know, in the lab to answer those questions. Anatomy of the nasal passages, the nose, the nares, the sinuses, what tissue is found there, the pharynx, nasopharynx, oropharynx, laryngopharynx, that where you have food and air traveling, it's going to be lined by stratified squamous epithelium. So you can read through that. The larynx, we had the epiglottis that regulates, the passage of material into the larynx or, over into the esophagus because it blocks off food from entering your airway system. The hyoid bone, of course, is superior. The larynx is composed of 3 cartilages: the thyroid cartilage, cricoid cartilage, arytenoid cartilages. The vocal folds, lined by the pseudostratified ciliate columnar epithelial tissue. So we've just kind of, the exercises help you just arrange it in a different way. You can use whichever method, either primarily studying from the PowerPoint notes or filling in those lab exercises. I think both work. You take the notes and you fill in the lab exercises, or you just use the functions that are provided with. All right, then we have in the trachea, we have our mucosa, submucosa, hyaline cartilage, and adventitia layers. So the mucosa is layered next to the lumen of the passageway, the pisket, its function, that there's goblet cells there to trap particles. The submucosa contains mucus glands and traps particles. That's why we have areolar connective tissue and simple cuboidal tissue there. We have the hyaline cartilage, we have the adventitia, all mentioned before. In the lungs, the main organ of respiration, contains airways and alveoli. These are the like conduction and respiratory conduction pathways. We have bronchi, bronchioles, alveoli. And so that discusses the histology, especially in the bronchioles. Love those because now we get to the area that is under autonomic nervous system control because of the smooth muscle there. But that also the mucosa changes from that pisket to simple cuboidal epithelium. The alveoli is your site of gas exchange, simple squamous epithelium. And so here, I just kind of help you kind of, you can use whatever method you want, but when it comes to the tissues that change or the tissue changes from where air enters to where we have gas exchange, it is primarily lined with pseudostratified ciliate columnar epithelial tissue, but there are a few exceptions. The oropharynx and laryngopharynx has stratified squamous epithelium. And then you go back to, to the pisket in the trachea and the extensive bronchi, you get to the bronchioles, simple cuboidal epithelial tissue, and the alveoli, simple squamous epithelial tissue. Okay. And now we are moving on to, some models. So here we have the half head. We looked at the tonsils at C, when we looked at the immune system, but we also have A that represents the sinus cavities. Technically you have the frontal and the sphenoid sinus cavities. You have the nasal cavity here at B, and anteriorly you would have the nares or your nostrils, right? Level with the tongue, C, you have the oropharynx, and then you have the laryngopharynx inferior to that. D represents the vocal cords or the vocal folds within the larynx. You're in the larynx when you have the blue hyaline cartilage to the anterior and posterior surface here. So that's some anatomy pointed out on that picture. Then we go to, here's the gray model that we looked at in the larynx. Here you have your hyoid, thyroid cartilage, We have the trachea below that. C is pointing to one of the cartilage rings. In between those rings, we have connective tissue. It gives the tube of your trachea a little bit of flexibility instead of having a solid pipe, for an airway. And D, we're going to come back to that in endocrine system, because that is your thyroid gland. All right. Then we go to the posterior aspect of the larynx. We have the epiglottis. B gives you a good picture of the arytenoid cartilages. C is the cricoid cartilage, and D is the fibula. So that's another. Here we have again that same model, large what are called cookie sheet models, where you can see A, which are those same sinus cavities. You don't have to name them individually, but know what the function of sinuses are. The nasal cavity at B, oropharynx at C, and D is the trachea. There are other things you could point to. You could point to the ribs and the intercostal muscles. You can point deep to that would be the pleural membranes. You could ask, hey, what is the function of, pointing to those ribs? What is their function in inspiration or expiration? Here is to the upper right-hand side of that same model, and then the lower right-hand side of that same model. All right, so now we're looking at our airway passage, right? We have the larynx at A. The hyoid bone would be above that, but it's not indicating that, but just to get orientation. Right, the larynx at A, trachea at B, C, all the way out to the little ends are bronchi. To the right, A represents the bronchiole, bronchoconstriction dilation, and B are alveoli. We also have the capillary network. We have little pulmonary arteries and veins and capillaries. So this kind of compares in a little chart the histology. Over to the left-hand side, we have the trachea, bronchi, bronchiole, alveolus or alveoli. Then we go, okay, the layers, the tissue found there in red, the functions over in red, in a chart form. And then you can either study it that way or fill in those exercises and then study from it. This is the histological slide of the trachea that I think is very, very good. So here you can see at A, the upper layer, adjacent to the lumen, so that's mucosa, pseudostratified ciliated columnar epithelial tissue. But between that kind of superficial purple stripe of tissue on top and the hyaline cartilage at C is the region of B, that is kind of a darker purpley staining region, I guess you could say. That's your submucosa. And so we have a lot of mucus glands, so that's simple cuboidal epithelial tissue. And the connective tissue is the areolar connective tissue is the most abundant. And then to the outside at D, where you can see adipose tissue there, that would be your adventitia, connective tissue layer. All right, so once we see C, which are the alveoli, and now, you can start to see the walls of the alveoli. You see the multiple dark staining nuclei that are on the circumference of it. That's telling you that those are individual cells, and those are the squamous cells that form these alveoli. So if you see alveoli, you're in the lungs. So now you have to identify, if the alveoli themselves, because that's a valid, identification. What is that structure? Alveoli. What's the tissue? Simple squamous epithelium. What's the function? Gas exchange. But the other tubes up here, those you might be asked to identify. B are the bronchioles, because we don't see any hyaline cartilage lined with simple cuboidal epithelial tissue, and where we have smooth muscle to bronchoconstrict and dilate. Epinephrine, norepinephrine, sympathetic nervous system, bronchodilate. That's the reaction, and that's the response. A is just pointing to a vein. We can often see other arteries and veins. You look into the lumen like you would to any other artery vein, maybe see if we can see some red blood cells in there, and that would help you with your identification. Remember, an artery will also be popped open with a pretty thick muscular wall. Right. So here we have another bronchiole, the same. I don't think this is the same slide. And you can see a little bit of the smooth muscle surrounding it. And so the fact that it didn't have hyaline cartilage and you do see some smooth muscle, that's your bronchiole. And C is another alveoli. These pictures are pretty good. These are great. Now, these are from actually from the University of Kansas, from their medical program. But look at those. Like, wow, you can see the hyaline cartilage all around the entire, virtually the entire circumference of that bronchi. Physiological control of respiration. It starts with the medulla oblongata. It sets the respiratory rate. We have chemoreceptors both peripherally and centrally in the body. Peripherally, they accompany those baroreceptors in the aortic arch and the carotid arteries. You're monitoring for changes. And then we have, let me just, before I move on, we also have central chemoreceptors within the brain. So it's monitoring for hydrogen and carbon dioxide levels in blood and cerebrospinal fluid. High hydrogen and therefore high carbon dioxide levels, or high carbon dioxide levels, produce high hydrogen ion concentrations. The response is to increase respirations or hyperventilate to release your carbon dioxide. If the levels are low, it implies that low carbon dioxide levels means the hydrogen ion concentration is decreasing. And so now the response to correct it is to hypoventilate, maybe hold your breath a little bit, to build up the carbon dioxide to increase your pH level. So, these are talking about, if the condition, what is the response of the medulla oblongata, essentially? If we have high pH values or low pH values, or high carbon dioxide values or low carbon dioxide values, how are we going to correct it? So if carbon dioxide is high, it means you're not getting the carbon dioxide diffusing in the lungs and exhaling it away. So what you want to do is exhale a few times, hyperventilate, and blow off the carbon dioxide that's there. And then if it's too low, then you want to slow down respirations, let some of that exchange slow down. Don't keep respirating and the gas exchanging. Cause the carbon dioxide to back up and increase the hydrogen ion concentration and decrease the pH. Proprioceptors around in the body, they monitor exercise, so that we can stimulate the medulla oblongata to start changing respiratory rate to accommodate, the physiological demands of exercising muscle that is impending. The sympathetic nervous system stimulation. Well, the stimulus is response to an autonomic nervous system fight or flight response. The production site for the change is going to be in the medulla oblongata, in the adrenal medulla, to release epinephrine and norepinephrine into the bloodstream. The target will be the bronchial smooth muscles, and the action is to dilate them. Right? So it would be an inhibitory response, essentially, for that muscle to contract less frequently and let it dilate slowly and increase the airway passage. Here we have pictures of what the ribs and the diaphragm are doing. So this is where we get, the information to what is the action of the intercostal muscles in the rib, the pleural membranes, the diaphragm. When we have inhalation and exhalation, you can go through that. Let's have them up there. We have breathing patterns. This is good to know. The process versus oxygen and carbon dioxide. When you are normal breathing, like you do hopefully right now, your oxygen levels should be normal and your carbon dioxide levels should be normal. Rebreathing, what we mean by that is maybe, when you have someone hyperventilating, you'll often say, breathe into a paper bag, not a plastic bag, a paper bag. And so as you exhale, you blow up the bag, so to speak, or you distend it. And then you inhale again. Well, when you do that, a lot of what you just exhaled is what you're going to inhale the next time. And every time you keep doing that, inhaling, exhaling into a bag that's kind of sealed around your mouth, you breathe in more carbon dioxide than just oxygen. Now, you don't want to do this in a plastic bag. There's a certain amount of that gas is going to be pushed out the walls of the paper bag because it's not completely sealed. But, when you do that, what's called rebreathing, the oxygen levels will slightly decrease and the carbon dioxide levels will increase. Holding your breath is the same way. So rebreathing, lets you have more exchange. Holding your breath means you stopped that ventilation process. So the oxygen levels are decreasing and carbon dioxide levels are increasing. Hypoventilation means you're breathing too little. So it's along the same, it's not the same, but I mean, all we're saying is that if you are hypoventilating and have normal ventilation, normal respiratory rates are 12 to 20 respirations per minute. If you're breathing, these cycles of respiration and the gas exchange are less than 12 per minute, then the carbon dioxide has opportunity to kind of back up into your bloodstream. So oxygen levels decrease and carbon dioxide levels increase. Hyperventilation or a ventilatory, let's say you had a question, patients, you're monitoring patient and their respiratory rate is 25 or 30. What's happening to the oxygen levels? What would you expect to see, their oxygen levels to be? What would you expect their carbon dioxide levels to be in arterial blood? Well, now the oxygen levels will increase to normal, or there's a maximum, and carbon dioxide levels will decrease because you're blowing off more carbon dioxide. So I would expect that the carbon dioxide level will be less than 35 millimeters of mercury. In the examples above of rebreathing, holding your breath, or hypoventilating, then I would expect my carbon dioxide levels to be a little to the higher range of normal, above 45, because the normal range is 35 to 45 millimeters of mercury. Here we have the carbonic acid cycle. You need to know that well. It's the respiratory system that can affect that carbon dioxide level to the far left. If we increase respirations, so you start to, hyperventilate, that carbon dioxide level will go down. So will the hydrogen ion concentration and the pH value goes up. If I slow down my respiration, now that carbon dioxide level, that component of this equation to the far left-hand side, is going to increase. And we know we have all the water we ever need, in our bodies. So it produces more carbonic acid, and then further over to the right, more hydrogen ions, which then decreases the pH. And knowing this and getting this down now is going to save you a lot of angst and anxiety, because that takes us to respiratory, effects to blood pH, acidosis or alkalosis, respiratory acidosis or alkalosis. On the other side of this equation to the right, bicarbonate and hydrogen ions, whether those are increasing or decreasing. Am I losing too much bicarbonate or gaining too much bicarbonate? Or am I gaining too much hydrogen ion or losing too much hydrogen ions? Those are the two variables when we name metabolic acidosis or alkalosis, which takes us down here. So you don't need to know a lot about this, but I would start thinking along these lines, because ultimately, yes, you're looking at blood saturation levels, you're looking at volumes of air, pulmonary volumes, functional volumes with patients. But a lot of it is what affect these changes in respiration or changes in the rest of the body metabolically. What impact does that have on the pH of blood? Because we need to maintain a normal pH level of 7.35 to 7.45. So below that, we have a chart that talks about the process. And these are the acid-base balancings that we kind of come back to on multiple occasions. But we can have respiratory or respiratory acidosis or alkalosis, meaning the etiology or cause of the change to the pH value you're looking at is some change to the carbon dioxide levels to from these particular regions, I will say, in sources that we're identifying in causes and etiology. It's essentially anything along the respiratory pathway, maybe brain injury that might affect the medulla oblongata directly, or all out cardiac failure. I mean, just asystole. You have no, blood, no heart contraction and no heartbeat at all. Right? Any of those issues fall under respiratory etiologies of respiratory acidosis, alkalosis. Everything else, changes in hydrogen or bicarbonate ions, increasing or decreasing, is going to be ultimately modified and adjusted, you know, by the kidneys. So we call those metabolic and acidosis and alkalosis. So respiratory acidosis, if we have respiratory acidosis, I think the easiest thing is to look at the pH first, because to be acidotic means you're below the 7.35 of the normal range of blood pH. And that is because we have higher than normal carbon dioxide levels producing more of those hydrogen ions. The oxygen levels generally have decreased, but it doesn't affect the pH. So what are the causes? Hypoventilation, emphysema, maybe pulmonary edema, really any of the and not just emphysema, but any of the three disorders, that are part of chronic obstructive pulmonary disease. Obstruction like choking, maybe brain injury that I described before, or all out, asystole. Have respiratory alkalosis. It means that the pH, so let's look at that first. I think that's easier, has gone higher than normal. And if it's a respiratory problem, it's because the carbon dioxide levels are too low. You're over respirating, perhaps. You're hyperventilating, and thus you're blowing off too much carbon dioxide. So etiologies would be hyperventilation, anxiety, which essentially anxiety, the gram-negative sepsis you're looking at, and fever ultimately cause hyperventilation. Doesn't have to mean, like 50 respirations per minute. It's just going to be higher than normal. When we have metabolic issues, which we will come back to again, not only during the urinary system, but also, especially in lecture, hopefully, where we kind of there's a whole chapter on acid-base balance, which I love doing. And I will probably do an extra credit seminar on this later in the semester. So when we have changes to hydrogen bicarbonate levels, really, because you can either lose too much bicarbonate, or have an increase of hydrogen ions, or you can have a loss of hydrogen ions and increase of bicarbonate. In any event, look at the pH first always. So when we say we have metabolic acidosis, it means, okay, the pH is lower than normal. That's why we're calling it, acidosis. So the pH is dropped below 7.35. But it's an etiology that is something other than these respiratory issues. So things like diarrhea, ketoacidosis, salicylate intake, which is too many aspirins, renal dysfunction. Those are just a few that can cause the hydrogen ions maybe to increase or lose bicarbonate ions. Metabolic alkalosis, the pH is higher than normal. And that's often due to prolonged vomiting, use of diuretics, and antacids. So again, we're either in prolonged vomiting, we're losing hydrogen ions, so now the pH value is going up. Or maybe we're taking in too many antacids, which are the buffers. Let's say you have an acidic stomach and now you've eaten way too many. And well, now you put in all this bicarbonate into the system to interact with the acids because you had, a burning stomach and the pH value is gone up, and you've interacted with a lot of and reduced the hydrogen ion concentration and thus increase the pH. So we have different ways of naming these. We'll get into this again, later, but that's kind of where we're going with the carbonic acid cycle ultimately. How does the respiratory system make adjustments to maintain an appropriate pH value in blood? It does so by varying the respiratory rate when needed. But if the respiratory system is not functioning great, because maybe a pulmonary disease like emphysema or chronic obstruction pulmonary disease, then chronic bronchitis, asthma, they're all part of the COPDs. then we generally have a buildup of carbon dioxide in the blood. And now, even though the respiratory system would like to change respiratory rates and try to address it, you have a disease process that doesn't allow you to do it. So this is where the urinary system will come in and help out and try to adjust the blood pH from that end. It's really very fascinating. All right, well, that's the end of this respiratory lab. I'm just thinking if there's anything else I need to add. I don't think so. All right, thank you very much.